Hi, everybody. I'm Eddie.
I'm Raj.
And I'm Ashwin.
And this is a warm welcome to this episode of Blood Cancer Talks, a podcast we are dedicated to hematologic malignancies, where we bring content experts who live and breathe a particular disease, or in this case, a particular modality, and focus on the latest advances in biology and clinical management. Please find us in your favorite podcast platform and rate and review our podcast, which helps us to spread the word. Today, we're very excited to talk about a new entity labeled by this group as CAR transgenic T-cell lymphoproliferative neoplasms, or post-CAR T-cell lymphomas, or CTTLN, the catchy acronym. And as I'm sure many of you have heard and read over the last sort of 18 months, these phenomena have been increasingly described now with a number of cases reported across some of the big journals in medicine. And as we're using CAR T-cell therapy and more frequently in hematologic malignancies, we're seeing these reports pop up. And a recent paper in the New England Journal, 2 of these cases were reported in the CARTITUDE-4 study of cilta-cel in relapsed myeloma, which was, of course, a randomized trial testing cilta-cel against standard of care treatment in lenalidomide refractory disease. And so today we're very lucky and honored to have Associate Professor Piers Blombery, who is the head of the Wilson Center for Blood Cancer Genomics, which essentially means he is in charge of the molecular hematology service for much of Australia and New Zealand here at the Peter MacCallum Cancer Center, and who's also the senior author on this New England Journal manuscript. And I should also add that Piers is also a lymphoma clinician and our local histiocyte a guru as well, another feather in his hat. But today we won't quiz him on histiocytes, we'll quiz him on post-CAR T-cell lymphomas instead. So before we jump into the topic, Piers, could you tell us a little bit about yourself and how you come to be a molecular hematologist?
Yeah, sure. Thanks, Eddie. So as you said, I'm a clinical and laboratory hematologist. So with my clinical hat on, I work in the aggressive lymphoma service here at Peter MacCallum Cancer Center in Melbourne, Australia. I also have a subspecial interest in histiocytic malignancies. I also run clinically the sort of genetic hematology substreams, dealing with a lot of the genetic findings of patients findings which we generate in molecular laboratory, and also run a large program in inherited bone marrow failure as well. That's mostly, a lot of that's fed from my other hat, which I wear, which is the head of molecular hematology here at Peter Mac. So we're a NATA accredited. So that's, a clinical pathology laboratory, which is pretty large. So we do about 15,000 samples a year of NGS testing exclusively in patients with blood cancer in both the somatic and inherited forms. Yeah, so that's what generally keeps me busy on a daily basis.
And what got you interested in molecular hematology? What sparked your interest in that area?
I did a science degree before medicine, and so I majored in biochemistry, so we did a lot of genetics and a lot of molecular aspects then. So I was always interested in molecular. I wasn't, to be honest, I just saw the writing on the wall and realized that the molecular-based hematologic malignancy was going to be a big deal in hematologic malignancy. Interestingly, when I was a trainee, the BRAF V600E mutation in hairy cell leukemia came out in the New England Journal, and at the time, obviously, I worked at a cancer center which deals with solid organ malignancy as well, and they had an assay for BRAF V600E, which they were using for melanoma. And as a sort of a research project, I repurposed that assay and just tested basically most of the patients in Australia with hairy cell leukemia at that stage. And that was the start of my explicit interest and research into molecular hematology as toe in the water to begin with. But then it went from there, and then I did some training in the UK, and then I worked as a consultant at University College Hospital London, where I was also involved in molecular over there. So it just snowballed from there. And then when I came back to Melbourne, Peter Mac started running the molecular hematology laboratory, and yeah, it turns out it's quite useful testing and quite informative, and it's just grown exponentially from then.
Yeah, I think it's probably going to continue to grow, but I'll come back to you more on that in a minute.
I like spreadsheets, so that's a natural love of spreadsheets, which obviously draws me to molecular hematology.
Of course. To set the stage on this topic, I think it's important that we break through the hype a bit, because there's been a lot of hype around this topic, I think perhaps led by the FDA. So to start with, can you tell us a little bit about the magnitude of the problem? How common are T-cell lymphomas post-CAR T-cell therapy, especially those expressing the CAR, and how commonly do we see it?
I'd like to be able to answer that accurately. I don't think I can. I think it's essentially unknown how common both T-cell lymphoma and CAR transgenic T-cell lymphoma is after CAR T therapy. I think it's really crucial in this arena to distinguish between T-cell lymphoma and CAR transgenic T-cell lymphoma, because they're completely different in their pathogenesis. They've got completely different implications, both regulatory and for the technology, and presumably will have different treatments as well. Post-CAR T-cell lymphoma, which if you like the broader hat, is we see occasionally. But we have to realize that patients with B-cell lymphomas are more likely to get T-cell lymphomas, and previous immunotherapy deficiency, they're more likely to get T-cell lymphoma. But the bullseye of what we're interested in talking about today, at least, and what I think what everyone's most interested in, is the CAR transgenic form. So that is essentially a lymphoma, and we can argue about whether lymphoma is the right term for it. We call it lymphoproliferative neoplasm, as to try and bring it back to pathology, because it's not lymphoma as we know it. Essentially, it's a transgenic malignancy. It's a transgenic neoplastic process. What's the instance of that? Very difficult to know. It won't be lost on us that the cases are coming thick and fast at the moment. There sort of seems to be one case every 2 or 3 weeks being published currently in either New England Journal or Nature Medicine. There's no sort of good surveillance program for it. It's quite sophisticated and difficult to prove that a patient's got CAR positive malignancy after CAR T therapy. So there's almost certainly going to be underdiagnosis. And also we have to acknowledge that in a patient with, if you think about diffuse large B-cell context and CD19 therapy, that when a patient progresses with an FDG-avid lymph node after CAR T therapy, there's a good chance that that patient has not a lot of options left, especially, and that may well not be biopsied and just presumed to be relapsed large cell lymphoma, when in fact it could be T-cell lymphoma or CAR transgenic T-cell lymphoma. I mean, the most evidence-based answer to your question is that there's been about 20 cases reported to the FDA out of 27,000 infusions. That's 0.1%, but we don't know, really. We don't know. We won't know until we have a prospective study where we aggressive with these things and actually actively look for it, because, as I'm sure we'll get into, it's going to be a continuum from monoclonal processes through to monoclonal processes causing symptoms, to monoclonal processes that are causing FDG abnormalities, to monoclonal processes causing something that's indistinguishable from lymphoma, to fulminant malignant acting neoplastic proliferations. And where we call that CAR transgenic lymphoproliferative neoplasm is important, and it's clearly going to be higher than we know at the moment.
And so do you think it's high enough to either factor into your decision making clinically about patients to give CAR T to, or to counsel patients who you're sending for CAR T about?
Look, I think it's important. Clearly, we should tell our patients about all the possible complications of the therapies we're given, especially ones which they're likely to possibly read about in the literature themselves. I think, obviously, at the moment, the benefits of CAR T for an individual in these circumstances receiving it are outweighing the risk of a 0.1% chance of CAR transgenic lymphoma. But it still behoves us to try and minimize that risk, know exactly what it is, and counsel patients appropriately. But currently, at the moment, it's clearly not a reason to stop the CAR T process, but it's a reason to look at what we're doing and how to make it safer.
Absolutely. And building on that question, if we look at CARTITUDE 4, you report 2 cases of CTTLN, which is out of 208 patients is about just under 1%. Do you think, obviously, we need a bigger sample size to accurately know what the incidence is, but do you think there's variability between different CAR products in terms of the frequencies or types of CTTLN? And is there any biological rationale for that in the types of CAR T products?
So I suppose the best answer we have for that is that overt CAR transgenic T-cell lymphoma has been seen now with, at least to my knowledge, cilta-cel, antigen, so CD19 and anti-BCMA, they're different CARs, and also the first CAR T transgenic lymphomas were seen with a piggyback transposon CAR insertion methodology, and that was a very small trial. That original description with the piggyback transposon, but that was in the order of 2 out of 10. So so clearly the CAR has some effect, but whether that's related to the construct of the CAR itself, whether it's related to the way it's inserted is difficult to know exactly. We need to remember also that we're giving these generally the CARs are restricted to different hosts. So cilta-cel is given to patients with myeloma, antigen is given to patients with large cell lymphoma, and there's lots of reasons to suspect that patients with myeloma might have a different susceptibility profile to getting transgenic malignancy compared to a patient with large cell lymphoma, whether it be age, pre-existing clonal hematopoiesis, relative immunopoiesis, pre-existing therapy, what therapies they go on to afterwards. It's early to say the one specific, outside the context of piggyback transposon, which there's reasons to suspect that the very high insertion site was actually quite a clear contribution. That we really have to work out what the contribution of the nature of the CAR, the nature of the host, the nature of pre-existing issues, and that will be the theme of all the cases described so far.
All right, so now to set the stage, we will start with a case. So I borrowed the case from your paper. We haven't seen any case yet at our center. So this is the first case. So just for the audience, it was a 51-year-old male with high-risk multiple myeloma, was double hit, diagnosed in December of 2019, and then was enrolled in the CARTITUDE-4 trial in November of 2021, which was a randomized trial comparing cilta-cel against standard of care, which was mostly Dara-Pom-Dex. Notably, before the trial, patient had progressed on VCD or Cybody, followed by MEL-200 autotransplant, followed by Len maintenance for 1 year. He was randomized to cilta-cel, received 2 cycles of bridging with Dara-Pom-Dex, followed by cell infusion, and achieved a MRD negative CR on day 100 with MRD negativity at 10⁻⁶. However, approximately 5 months post-infusion, he developed a rapidly growing nasophacial plaque, which was FDG avid, and biopsy showed a monoclonal T-cell infiltrate, which was CD2 and CD3 positive, but was double negative for CD4 and CD8. At that point, the patient was diagnosed with PTCL-NOS, peripheral T-cell lymphoma not otherwise specified, as per the current diagnostic criteria. Piers, can you tell us, first of all, give us a big picture overview of the translational studies that your team performed on this case to get to the bottom of the origin of the T-cell lymphoma?
Yeah, sure. We basically just did a lot of genomics, which is what I do. And I think it's important to emphasize that we called it was diagnosed as PTCL-NOS as per current diagnostic criteria, because there's not, and you can argue actually didn't actually fit PTCL-NOS from current diagnostic criteria, because the current diagnostic criteria is silent on transgenic neoplasms. But clearly that was the best bucket to put it into in the first instance. It was a clearly immunophenotypically abnormal T-cell population, which was CD4 double negative, morphologically infiltrative and monomorphic under the microscope. So it fit what we would know previously as PTCL. As PTCL-NOS. So we established with monoclonal through standard, TCR sequencing. So when you were dealing with a monoclonal population, you'd be surprised at anything else, given it was double negative by 4 and 8. And then I suppose the critical part after that was essentially showing that all the cells were CAR expressing. We did this in a few ways, by immunohistochemistry as well as by flow cytometry, to prove that the CAR was very highly expressed in the tumor. So clearly they were all CAR positive. So that was the first indication that we were dealing with a CAR transgenic process. And I'm choosing my words carefully here, because I think because the words matter in these circumstances, what we call things. So this monoclonal process was expressed in the CAR. We then did whole genome sequencing to get an idea, first of all, of the nature of the CAR insertion, as well as the other associated abnormalities. And I suppose in no particular order, we found a single CAR insertion site into the 3' UTR of PBX2, and that was through the structural variant caller, etc., reassembled the CAR sequence into the 3' UTR of PBX2. There was only one insertion site, which was important because the previous, as you might know, CAR T lymphomas, which we described with the piggyback transposon, actually had about upwards of over 15, 20 insertion sites. Yeah, that's because piggyback transposon is a lot more efficient, and people will know who in the CAR T industry that you sort of aim for it. You really want about one insertion site per genome, and so there was only one insertion site. So that was the important thing to see, that it wasn't the same as the previous ones we've seen, where the CAR had really peppered the genome through and caused lots of disruption. It was just disruption in PBX2. We noted on whole genome sequencing that it was a relatively quiet genome, so it wasn't super hypermutated. There was no overt mutational signatures. There were a few what I would describe as non-specific variants seen. There was a duplication in NFKB2 and a few other things which is just non-specific for any particular process. But the other thing that caught our eye was that the patient had TET2 as well. We presume biallelic TET2s based on the variant allele frequency, so mutations inside the tumor, inside the neoplastic process. So based on those, when we saw those, so that was a summary of the whole genome. So insertion
so Pierre, just to clarify, this is not, you didn't identify TET2 based on whole genome sequencing, it was targeted sequencing.
No, it was in the whole genome. We did about 10 things at once, and so they were all coming back. But yes, the whole genome. There were 2 TET2 mutations in the whole genome sequencing, as well as we did target sequencing as well, which found it as well.
And what was the depth of the whole genome sequencing?
So it was the tumor about 100x, and the germline about 50x. So it was relatively deep whole genome, but the target sequencing is about 1000x. But they were very high VAF, so they were both at about 40%, consistent with them being biallelic. One TET2 was quite interesting for the genomic nerds out there. It's actually in a non-canonical transcript upstream in a non-coding exon, causing splicing abnormalities, but that's not the point of it. But suffice to say, one was a relatively typical TET2 variant. So given the fact that this, as people will know, or if people don't know, TET2 variants are seen in T-cell lymphoma, particularly in a subtype derived from T follicular helper cells called angioimmunoblastic T-cell lymphoma. And in fact, as I say to my registrars and medical scientists, is that angioimmunoblastic T-cell lymphoma is actually not, it's not best thought of as a T-cell lymphoma. It's thought of as the T-cell manifestation of clonal hematopoiesis. So you get a TET2 mutated hematopoietic stem cell. If that causes a myeloid malignancy when it's in the myeloid compartment, and in the T cell compartment, it manifests itself as AITL, and in fact, and in the B cell compartment, it manifests itself as what people call large cell B cell diffuse B cell transformation, or AITL. But essentially, all those things are just manifestations of ultimately the ultimate manifestation of TET2 clonal hematopoiesis occurring in the hematopoietic stem cells. So we went back and looked at the patient for whether this TET2 was present previously, and we actually used duplex sequencing from TwinStrand, so highly sensitive sequencing, and we managed to get a vial of CD34s from his previous autograft, and were able to find the TET2 variant in the CD34s from that was taken two or so years before he was exposed to CAR T. So he clearly had pre-existing.
And what was the VAF at that time?
Oh, extremely low. So yeah, it's extremely low, 0.01%.
Okay. And that particular small TET2 clone growing in size, becoming a biallelic, I think that's pretty impressive.
Yeah, and it turns out that's going to be, if you look at all the descriptions coming out, that's going to be the central to the pathogenesis of a lot of this, is that you've got a pre-existing TET2 clonal hematopoiesis present at, it doesn't have to be present at low level. Presumably it can be present at high level as well, but you collect the T cells, which, when we know that TET2 clonal hematopoiesis skews into the T cells in a significant proportion of patients, you collect those cells and transduce it with the CAR, and then that's the basis of, I suppose, that's the first inverted commas hit, if you like. And so we managed to, and we also showed that it was present in the product by definition, given the fact that it was present beforehand and present afterwards. The other thing we found, interestingly, was a variant in the patient's germline in JAK3, which was this variant keeps popping up throughout my career in various different forms, but it's the JAK3 V722I, which is it's an interesting variant. So It's a relatively common germline polymorphism, so it's present in about 0.5 to 1% of the healthy population. So in NOMAD, it's present in a reasonable proportion of the healthy population. But the first time I clocked it was in the context of a patient with breast implant-associated anaplastic large cell lymphoma, when this patient had a patient who developed anaplastic large cell lymphoma in the context of breast implants, had this JAK3 V722I in her germline. And this V722I has actually is shown to transform T cells when you put it into cell line models, and it shouldn't be lost on us that the current pathogenesis of breast implant associated anaplastic large cell lymphoma is felt to be chronic stimulation by bacteria and a biofilm of chronic stimulation of T cells, which then overtly become malignant, like the T cell equivalent of marginal zone lymphoma. And so we've got a patient, to stay in the breast implant associated context, we've got a patient with a JAK3, with a germline variant, which is known to transform T cells, i.e. make your T cells a little bit hyperactive and mischievous, so to speak, not to anthropomorphize T cells. And then in the context of chronic antigen stimulation, have the ability to turn into an overt lymphoma. That's not dissimilar to what we're seeing here, where you've got this patient with a JAK3 V722I who then has had chronic stimulation of the T cell, not through bacterial stimulation, but through a transgenic receptor to be stimulated. Look, that's all completely speculative, but I note it because it's the second time, and also it's relevant because we actually, you'll see in the treatment, we actually gave the patient tofacitinib, which is a JAK inhibitor, and that was the rationale. That was the rationale for that, just in case it was contributing to the pathogenesis. So look, that's the summary of the genetics we did. But insertion into the PVX2 3' UTR, biallelic TET2s, some non-specific variant changes, relatively quiet genome, and a germline JAK3.
Piers, I have a couple of follow-up questions. I think so very comprehensive summary of all the translational studies here. So first,
you mean too long? Is that what you're saying? You're saying too long? Is that what you're saying?
No, it was perfect.
Is that the polite way of saying too long?
No, it was perfect. It was perfect.
The less polite way is we just edit it down and don't tell the guest. So that's the polite way for sure.
Yeah. In the clinical context, if we encounter a case like this, and if there is CAR positivity on the T cells, does that automatically mean that the CAR T cell therapy caused the lymphoma? Does it prove causality if there is CAR positivity on the malignant cells?
Yeah, people ask me that question a bit, and I find it difficult to answer that question. If you've got a lymphoma that's derived from a transgenically altered T cell, then the null hypothesis is that the transgenic process has caused the CAR T cell lymphoma. So it's more, in my mind, it's more I don't see how you can argue that it's not the overt cause of it. Now, what the actual contribution of various factors around it is, but I think it's unlikely that it doesn't make a lot of sense to me that you would presume that in the absence of the CAR therapy that the patient was going to develop a T cell lymphoma from that particular clone. I don't think makes any real sense. So yes, it's the ultimate cause, but I suppose the approximate question that you and a lot of other people are interested in is, what is the actual contribution of the CAR T insertion, i.e. insertional mutagenesis, to the actual process? Is it something because that has implications for the technology we use and whether we start and how we focus on trying to prevent it. If it's a general non-specific thing that happens when you just stimulate a bunch of T cells, then that's going to be very different to if it's 100% of the time overtly caused by insertional mutagenesis. Then there's a very different way to stop those two scenarios. Yeah, once again, it's a difficult question to answer, but yes, it wouldn't have happened without it. But what the contribution is, I'm sure we can get into about what the actual contribution of all those factors are.
Yeah, then another thing that comes up, actually, I was discussing with one of my colleagues yesterday, and they were discussing about checking potentially for chip mutations prior to giving cilta-cel, given that we have now these two cases with TET2 mutations in them. Specifically because with cilta-cel, as we had discussed before, out of 200, 2 patients developed it. It's 1% seems quite high, non-trivial at least. Would you check for chip mutations or do a whole targeted myeloid sequencing before cilta-cel in all patients? And if you find a chip mutation in a patient who is otherwise not high risk or has other options for myeloma, would you rather choose an alternative treatment and not do cilta-cel in that patient?
So no, yes and no, which is in our case, this TET2 wasn't overtly detectable by any practical mechanism, right? So it was present at super low level, and no one's got looking at 0.01% VAF beforehand. It won't be lost on us that the incidence of clonal hematopoiesis over the age of 60, if you look hard, I mean, it's essentially ubiquitous. If you look at it a sensitive enough methodology, it's essentially in everyone. It's actually in everyone probably by the age of 25. It's actually probably in everyone from work from the Cambridge group present actually embryonically. And so it's clonal hematopoiesis exists in everyone. So if you look hard enough, and the incidence of even overt clonal hematopoiesis in myeloma, in patients with myeloma, is probably going to be, once you consider all their previous therapy and various IMiDs they've been on, which may be associated with clonal hematopoiesis, it's probably in the order of 10, 20%, 30%. And we don't see 20% of patients getting CAR transgenic lymphoma. So it's obviously not enough in and of itself. It's, as I say, it's probably part of the story, but it's if you like, it's the groundwork that may or may not be required. But in terms of checking for it specifically, no. And I wouldn't check for it specifically to try and work out whether the patient is at risk of getting transgenic lymphoma. However, what's more common than CAR transgenic lymphoma post CAR T is myeloid malignancy. Now, once again, whether that's related to the immunosuppression, previous therapy, the inflammatory milieu afterwards, and now there might be a reason to check clonal hematopoiesis before then, in the anticipation that with a lot of patients with CAR T, they end up being cytopenic at day 28 and getting marrows and getting NGS to exclude TMN, and so you end up doing panels, doing target sequencing on them anyway, and so it's probably the most useful reason to look for clonal hematopoiesis up front is to inform the potential tracking of clones afterwards to look at TMN and the possible development of TMN, therapy-related myeloid neoplasm, let's say TMN.
Just for our audience, how common is therapy-related myeloid neoplasm post-CAR T-cell therapy?
Again, unknown. It's probably in the order of 1 to 5%. That's what's coming out as around the middle estimate from a lot of studies. Yeah, so it's not uncommon, and look at therapy-related myeloid neoplasm is very difficult to diagnose post-CAR T. Prolonged cytopenia, it's very difficult to make a diagnosis in someone, a multi-treated patient with morphological dysplasia. They've all got clonal hematopoiesis. Virtually, I don't think I've ever seen a marrow without clonal hematopoiesis post-CAR T, especially in that when they're very hypocellular, because presumably the only things that are surviving in that context are hardy clonal hematopoietic, derived hematopoiesis. Making it, you often need to wait till they overtly start getting blasts or unequivocal morphological dysplasia plus cytogenic abnormalities to make that full diagnosis of TMN. But it's more common than, well, currently it's likely more common than CAR transgenic T-cell lymphoma.
Now we'll briefly touch upon the second case in your paper, which is a 54-year-old female with high-risk multiple myeloma who got VRD-based induction therapy followed by autotransplant, and then followed by LEN maintenance. Unfortunately, the patient was relapsed and was subsequently enrolled in CARTITUDE-4 and randomized to cilta-cel. Patient achieved MRD negative CR. Unfortunately, ultimately developed a CTTLN 16 months post-infusion. So at a clinical and a molecular level, what were the similarities between these two cases?
Yeah, so they were strikingly similar. So clinically, even just the involvement of skin, it was clearly prominent in both cases. That could just be chance. T-cell lymphoma classically involves extranodal sites more frequently. It was notable that they both had skin involvement. Another clinical aspect that was similar between the two, which we haven't really talked about, but we don't really know what the contribution is, and this is going to be confounded, but both patients had infections that were proximate to their development of T-cell lymphoma. One patient had COVID-19, the other patient had parvovirus. now patients get infections with my post CAR T. It'd probably be difficult to find a patient that didn't have had an infection, but it's notable that they both had viral infections before. We looked at the CAR T sequence, whether we speculated that maybe the CAR sequence had specificity to COVID-19 or to parvovirus, the native T cell receptor. That didn't look to be the case. Yeah, but to be honest, those tools are pretty blunt in terms of working out specificity of T cell receptors. But anyway, that was common between the two. And then clearly this patient also had a TET2 biallelic TET2 mutations as well. And the insertion site of the second case was into ARID1A, which is, now people in the T cell lymphoma field may not have heard of PBX2 in the first case, but ARID1A is a pretty well described tumor suppressor, which is a chromatin remodeler, which is with loss of function mutations in patients with T cell lymphoma. I think we're a bit closer to the heat source there in terms of insertion overtly into ARID1A being a overt contribution. Now, we weren't able to conclusively show that through expression of RNA, but it was notable that was in ARID1A. Now, and we need to remember also that these CARs insert into near transcriptionally active genes. It's pseudo-random, it's not completely random. So there's a likelihood that they are going to insert into biologically relevant genes, or at least near biologically relevant genes. But ARID1A obviously is piqued our interest. Yeah, so very clinically very similar, molecularly very similar. Yeah, pointing to a sort of clearly shared pathogenesis.
Were both cases at Peter Mac, or
one was, and the other one was from France, our French collaborators.
At a follow-up, based on these translational studies that you performed on these two patients, can you summarize what are your arguments for and against a cause and effect relationship between cilta-cel infusion and subsequent T-cell lymphoma?
Yeah, so I think, well, as I said before, I think it's the ultimate cause. I mean, there's transgenic malignancy. But once again, to break down the contributions, which are not necessarily going to be the same in all patients. Some might be more important than others, and I think we can talk about other cases in literature where it's clearly more overt that insertional mutagenesis played a role. But there's possible germline predisposition, i.e. JAK3 V722I. There's pre-existing clonal hematopoiesis getting transduced. There's contribution possibly of proximate infection. Now, whether that's through a general just expansion of T cells or specifically of that clone, we don't know. And then insertional mutagenesis. Now you can wind the dial on each of those four as to how much they're contributing to each case, but it's just a classic model, multi-step model of developmental malignancy, which we see across cancer, whether it be blood cancer or solid organ cancer. Is you start off with some abnormality, germline or not. If not germline, clonal hematopoiesis. You cause those cells to rapidly to proliferate. Naturally, the cells that are most fit will survive. They develop mutations as we all are developing mutations, and in a proportion stochastically, some of them are going to be in oncogenes, tumor suppressor genes, which are going to go on to overt and create overt malignancy. So that's the cause, that's the model. It's just an accelerated form because of various factors like clonal hematopoiesis, the vigorousness of the expansion, and that's also the basis of what therapies or what we might do to actually try and decrease the incidence of these.
I'm just trying to understand the contribution of this JAK3 single nucleotide polymorphism, how it plays in this entire lymphomogenesis. Does it, right now, the reason I ask this question is right now we do not include JAK3 in any of the clinically actionable myeloid panel. We do not include JAK3, only JAK1 and JAK2 is tested. JAK3 is never tested. So should we be including JAK3 in our testing panel to see who are these patients who are increased risk of developing as a first hit followed by second hit, which is a clonal hematopoiesis?
Yeah, I mean, look, JAK3 is useful to test for anyway because it's relatively common in T-cell lymphoma, and one of the most common questions we get asked in molecular lab in the lymphoma context is, does this patient have T-cell lymphoma? Is it reactive? What sort of T-cell lymphoma do they have? And in that context, JAK3 is quite helpful because you commonly get activating mutations in JAK3 in various T-cell lymphomas and TALL and a few other things. But looking for this germline polymorphism. I don't think there's no clinical relevance at the moment looking for this germline polymorphism. In fact, we see it obviously in 1% of the patients we test. And so it's difficult to know what to do in that circumstance when they're, I think this is speculation, but I would guess that, don't forget that V722I has been described somatically inside T-cell lymphoma as well, and it's an activating mutation in cell line models. So it clearly causes activation of T-cell in T cells. My guess, my assumption, my speculation is that germline polymorphism may well be a predisposition to autoimmune disease / I think if we did a big enough study with the V722I, it would be a good candidate for correlation with general T-cell dysfunction, that not necessarily T-cell lymphoma, but once again, that's not in any actionable way that I suggest people go out and get tested with the V722I. But I suspect if we were to get a cohort big enough, that would be a very interesting thing to look at.
We should just send it in MGUS patients just to create a bit more chaos in the MGUS clinic. Or in the CCUS clinic is probably more appropriate. But the real question I want to ask you is, going forward, if a clinician was seeing their patient after CAR-T, such as cilta-cel, and they found a suspicious cutaneous lesion or even lymphadenopathy, and biopsy suggests a T-cell infiltrate, what would the next steps, do you think, in clinical practice be in the workup to inform the treatment, or if the patient needs treatment?
Yeah, sure. Importantly, implicit in your question is that the most important thing's been done and the patient's had a biopsy and it's not just been assumed to be relapse of the original malignancies. Look, on the presumption that you've got a monoclonal T-cell process post-CAR-T, including the next, once you've demonstrated it's monoclonal either by T-cell rearrangement or by immunophenotypic abnormality, clearly the next most important thing to do is to somehow demonstrate expression of the CAR or presence of the CAR or not inside the T-cell lymphomas. There aren't a lot of easy ways to do that. And practically, I think a lot of centers around the world have made their own assays, whether they be molecular for them or immunohistochemically, digital. Some people have got digital PCRs for some constructs. If you run a relatively broad panel, especially one that includes some of the components of the CAR, CD28, etc., then the structural variant caller will call it quite easily, because the structural variant caller will recognize that there's a high level of CAR in there. But that's probably even less practical than having a digital PCR. Essentially, however you do it, demonstrate that the CAR is present or not present in the T-cell infiltrate, and then whether you're dealing with CAR transgenic T-cell lympholytic process or a case of coincident T-cell lymphoma in a patient who's had either multiple myeloma or DLBCL or whatever they had their CAR for.
All right. So now I wanted to quickly discuss the case from Memorial Sloan Kettering Cancer Center that was also published almost simultaneously, I think, in New England Journal of Medicine. This was a case also post cilta-cel, a patient developing CAR transgenic T-cell lymphoma in the duodenum, not in the skin. So it was a duodenal biopsy showing a CAR transgenic T-cell lymphoma. And interestingly, the translational studies that were done showed integration of the CAR vector within a canonical tumor suppressor gene, TP53 in this case. So can you give us a brief overview of the molecular aspects of this case? And I was curious as to whether are you convinced that this is an example of insertional mutagenesis leading to a CAR transgenic T-cell lymphoma in this particular case?
So look, this is a very instructive case. This is, in my opinion, overt insertional mutagenesis into a tumor suppressed gene, the p53. I'm not sure that many people are going to argue that having a CAR inside disrupting the guardian of the genome is likely to be unrelated to some sort of downstream pathogenesis. The authors did p53 immunohistochemistry, showed absent expression as well, so it clearly disrupted the expression. So I think this is a good example of what I was talking about with the dial going up and down. If you think about the three cases we've spoken about, PBX2, unknown contribution, how that's contributing to ARO1A, suspicious, but we weren't able to do RNA. Here, insertion into a tumor suppressor gene, absent expression of p53. Like that's the spectrum of how much that can be, I suppose can contribute. Now, I'm not saying the other two aren't contributing, but this is the most overt, in my opinion, evidence of insertional contribution. Interestingly, this patient had a DNMT3A overtly present, and going to your previous question is that this patient overtly had a DNMT3A detectable in their peripheral blood, and that actually wasn't the cell which the T-cell lymphoma was derived from. That's a case where it just proves that there's not a lot of point detecting clonal hematopoiesis beforehand and avoiding CAR T, because this patient actually, it came from at least as far as my reading of the paper, it wasn't obvious that there was a clear contribution of at least not TET2 clonal hematopoiesis beforehand. This might be an example of no germline, a lot of insertional mutagenesis, not a lot of contribution from clonal hematopoiesis, and then contribution of the other infections and chronic stimulation unknown. I think it's pointing to, all these things are pointing to a multifactorial and really patient-specific contribution of factors to developing these malignancies.
Yeah, it's interesting that even within the CTTLN, there is so much heterogeneity as to the pathogenesis can be different in two different patients, even when they're developing T-cell malignancy after cilta-cel. So are there any...
that also explains that's going to be manifest in the diverse clinical nature of them. Well, some of them are rapidly, so the piggyback transposons, some of those were very aggressive, and the patients unfortunately died very early. Some of them act more like chronic, almost cutaneous T-cell lymphoma, waxing and waning, and those things are going to ultimately be driven by the molecular pathogenesis, plus or minus how much they're still receptive to ongoing antigen stimulation. But and it also was going back to what I was saying at the start, is about this, as well as being a different contribution to the pathogenesis, there's going to be a spectrum of patients who have a monoclonal expansion of no doubt if you look hard enough, there'll be clones that take off and are monoclonally dominant inside the patient in the inverted commas normal response in CAR T, which then go on and either atrophy and die off, or go and get selected for further. And where we draw that line between when we decide something's a monoclonal process of no clinical significance versus overt lymphoma is important, and not dissimilar to what Eddie was saying before about MGUS clinic. It's where we draw the line between a pre-malignant process which we need to be alert but not alarmed about, and overt lymphoma which we need to jump in treatment is going to be the key, and ultimately determine both molecular workup as well as clinical manifestations.
And what do you see as the sort of future for the field in looking to better understand the pathogenesis and ideally prevent the pathogenesis of these CTTLNs?
Yeah, to roll call across the pathogenesis we talked about is, well, germline, we don't know. That clearly more research into whether germline predisposition, whether there are patients who are overtly predisposed to getting through some sort of germline predisposition to getting T-cell malignancy. I think that's speculative at the moment, but it's worth researching. Insertional mutagenesis, I think people are always surprised to hear that if the patient asks, where does the CAR insert, or people who don't, physicians or scientists who don't know about CAR Ts, where the CAR insert, and we say randomly across the genome, that doesn't necessarily seem to be wouldn't necessarily be the best design if you were trying to stop someone developing lymphoma or a transgenic malignancy. So clearly guided insertion to a place we know is safe and it has the lowest risk of downstream abnormalities is important, and there are various technologies that are coming out to do that, CRISPR, etc. Then clonal hematopoiesis, whether we test for that, whether we monitor that, whether that becomes part of the CAR T QC product, whether we look at whether there's overt clones in there that are transduced, and whether we select out these pre-malignant clones in the process. Like, I'm thinking years in advance here about technologies we don't necessarily have yet, but things that you would do to make it safer. And then people often talk about kill switches and things in CAR Ts, so ways of turning off CAR Ts. It's worth noting that I don't think we've actually got any evidence currently, and this is something we need to research probably quite urgently, is how much that T cell is actually still dependent on the CAR to survive. I would suspect that, at least in our patients with enough abnormalities, that the CAR is possibly not contributing that much to the growth signal of the T cell anymore. Certainly in our patients, they were BCMA negative, so they weren't getting over antigen stimulation. Whether those T cells, whether those CARs have become autonomous or not, we don't know. That will be important to research, and that would be then a rationale why a kill switch will be useful in that context. But if I had to speculate, I would say that my suspicion is going to be that once you get to an overt CAR lymphoma with enough genomic abnormalities, the actual ongoing dependence on CAR signaling is probably unlikely. there's a suite of things to do, and I think it's behoved us to look at all of them really, and to work out how to make this clearly life-saving, important breakthrough in cancer as safe as possible.
I'm about to round up, so if you have something else you want to ask,
Okay.
then now's the time.
So, since I want to briefly talk about that fascinating case study you published in New England Journal of Medicine about UBTF, tandem duplication AML, and the menin inhibitor, since I'm a myeloid person in the podcast, can you briefly talk about that case and what we learned from this particular case?
UBTF tandem duplication AML is such an important AML to recognize. It's originally described by Jeff Clow and St. Jude's group. It's part of the larger NPM1 HOXA9 class of acute myeloid leukemia. It occurs in young people, primarily children, young adults, although we have seen it all the way up to people in their 70s. So it can occur throughout, but clearly it's enriched in children and young adults. It's very poor prognosis malignancy. It's often relapsed post-allograft. They often have primary refractory disease. And so for all those reasons, it's super relevant to identify, both because it's screaming out to recognize and treat it differently. And what we noted and what Jeff Clow's group noted is that it's part of the HOXA9 and the HOX dysregulated gene NPM1 KMT2A rearranged, all of which are being currently furiously treated with menin inhibitors as a very effective agent at inducing response and we were lucky enough in that patient to get menin inhibitor for that patient's refractory disease, and he responded. He almost went to a complete response with menin inhibitor. And then the other thing we noted then is, unfortunately, he progressed on the menin inhibitor and he developed the typical MEN1 resistance mutations, which are menin inhibitors. Biologically, that was important to show that it's in fact it was an on-target effect of the menin inhibitors. So I think from that case, it's a single case, but I know anecdotally of multiple people now giving menin inhibitors for UBTF AML. It clearly induces responses. Giving a single targeted therapy for acute myeloid leukemia is never going to be the way of keeping a long-term remission. So we need to work out the best way of using menin in NPM1 and KMT2A rearranged as well. But UBTF, and clearly one would think it would be in the upfront setting, somehow combined into the upfront setting, because a lot of these patients are in fact primary refractory, and as you all know, it also co-segregates with FLT3 abnormalities as well. I can imagine upfront FLT3 inhibitors plus menin inhibitors plus chemo, and then I think we'll end up allografting a lot of these patients because they're higher risk. But whether there's a rationale for then menin inhibition after allograft or as a sort of maintenance therapy makes more sense than relying on it as a single therapy in the overt leukemia stage, where in no cancer does a targeted therapy last for in almost no blood cancer, CML excluded, does a targeted therapy last for any longer than 6 or 12 weeks when given as a single agent in the blood cancer like AML, which is heterogeneous and proliferative. But so high level summary, extremely important to identify those. They respond to menin inhibition both in vivo and in vitro. Jeff Clow's group also showed that they did an in vivo model. We need to do more research. How do you recognize it? So it's very easy to recognize. It's a capillary electrophoresis. It's an ITD. If you can do FLT3, it's just different primers and you'll get the majority of them. You may get all of them. There are some bigger ones which are partial tandem duplications, but it's a relatively straightforward capillary electrophoresis, and we and other people have published our assay. Make sure you're testing for it.
Do you use right now routinely in clinic?
Yep. So it's on our targeted panel. So there's actually other exons are involved in UBTF. So there are rarely other exons involved. So we actually target all exons of UBTF on our targeted panel. So, and we find it, and again, it's also important to not just look for it in kids or just look for in the context of FLT3, because as I say, we've found it in 7-year-olds with AML unexpectedly, and you really have to throw all assumptions out the window when you're doing molecular testing and just unbiasedly test everyone for everything. That's my approach.
I think this is a fantastic case which highlights, I think there are probably more and more mutations or genomic abnormalities which overexpress HOXA9, which potentially can be targeted using menin inhibitors.
Yes, and there's no doubt will be other family members as well. That it would actually be best to be we're one step removed there from the pathogenesis, right? We're NPM1, KMT2A, UBTF. Obviously, we're one step removed from the actual transcriptional profile. It would would presumably be the best to actually have a robust transcriptional recognition, and then regardless of whether we can detect a whatever it might be, NUP98, or that the patient responds to menin inhibitor. But gene expression profiling has been difficult to get into the molecular pathology laboratory, which is the subject of another podcast.
All right. Thanks, Piers, for the great discussion today. Now I think I can make sense of these publications on CTTLN, and also glad to have a small bonus discussion on the menin inhibitor as well. All right. So thank you very much for coming on, and hope to have you back again for discussing more on molecular hematology.
Pleasure. Thanks for having me.
Thank you.